Hello girls, now let's learn about total RBC count. So we know that blood is composed of cells and one type of cell present in the blood is RBC. So we are going to determine the total RBC cells present in the blood sample. So RBC cells are also known as erythrocytes. They are the abundant cells in the bloodstream. They contain hemoglobin, and the main important function is they carry oxygen throughout the body. Any disruption of RBC cells in its quantity, shape, size, structure or life cycle can therefore affect the oxygen carrying capacity of the blood. So basically when we talk about the morphology of erythrocytes are non-nucleated biconcave discs. The cells possess on the surface a number of antigens which are responsible for blood grouping, as we have discussed earlier. The function of the red cell is to transport oxygen through hemoglobin in a functional state. Lifespan of the red blood cell is 120 days. And the erythrocyte is performed to determine whether the person has anemia or polycythemia. So almost the clinical significance is the same, to know whether the person is anemic or he is suffering from a condition called as polycythemia, where the number of red blood cell is more than the normal range. So when we discuss about the normal values of RBC cells, the normal values of RBC cells in case of male is 4.5 into 6 into 10 to the power of 6 cells per cubic mm, or 4.5 to 6 million RBC cells per cubic mm of blood. Similarly, female, it is 4 to 4.5 million cells per cubic mm of blood. So why should we carry out RBC count? So basically when we talk about why should we carry out RBC count, it is mainly to know whether the person is in its normal state or whether the person is mainly suffering from a condition where there is decrease or increase in the RBC cells called as anemia, or whether he is mainly present in a condition called as polycythemia. So whenever we try to know about all these conditions, we try to find out the normal health status of an individual. So how do we count the RBC cells? So we have a equipment which is called as hemocytometer. The hemocytometer is mainly a meter which is used to count the cells of the blood. So basically manual method is using a hemocytometer, where the blood specimen is diluted 1:200 with the